Yes, thank you. Now we'd like to introduce Dr. Luciana Balboa. Here she comes. Come on up. Price. And this is accelerating BCG efficacy, targeting dendritic cell migration in tuberculosis immunization.
Okay, great. Okay, great. Forward is this one, I guess. Okay. Okay, let's do. Okay, that's it. Okay, well, thank you all. Thank you for this great opportunity. Can you hear me? Yeah. Okay, thank you for this great opportunity to share our work, which has been awarded a Validate Grant Priming Grant that has been recently funded. So, forward, this one, El Grande. Okay, thank you. So we all know that tuberculosis still remains a global health problem, despite the fact that its approved vaccine, the BCG, has been in use since 1921. And in fact, the protective efficacy of BCG still remains controversial, as it varies between 0 and 80% in different populations and geographical regions. So despite the global use of BCG, we still don't have a clear immunological correlates of protections, which of course hinders the development of more effective vaccines. And there are at least 2 studies that clearly highlights this challenge, and I will briefly summarize them. So this first study demonstrates that neither the magnitude of T-cell response nor the cytokine profiles at 10th-week post-vaccinations can predict TB protection over 2 years in children. And there is another study in which the authors could identify potential correlates in infants, although such as, for instance, BCG-specific T-cells secreting interferon gamma cells that were associated with a decreased risk of TB, although the protective effect appeared to be limited to the first 6 to 12 months of follow-up. So these 2 studies underscores the fact that defining correlates of protections is complex, and that traditional static measures of immunity, such as cytokine levels or cell counts at isolated time points, cannot be sufficient. So to address this, what we propose is a novel correlate based on investigating the kinetic of the activation of the immune response. So what we propose is to test whether the speed at which the adaptive immune response is activated following immunization can serve as a predictive indicator of protection. So, but first we need to understand how can a fast adaptive immune response be achieved, specifically in the context of BCG immunization. And there are 2 main actors that need to be achieved in order to get an efficient, competent T-cell priming. So we need dendritic cells reaching the draining lymph nodes, and we also leave BCG within the draining lymph nodes. But how can leave BCG get or reach the draining lymph nodes? Well, of course, it is mostly accepted that skin dendritic cells are the main populations involved in this trafficking of BCG from the skin to the draining lymph nodes. Okay, the other one, sorry. This. So also free BCG can also travel from the skin to the draining lymph nodes, although it has been demonstrated that this free BCG cannot lead to significant T cell priming. And in another study, it has also been demonstrated that at least during the early stages post-immunizations, neutrophils can also have a role. In a very specific model of immunization, it has been demonstrated that neutrophils are the main population hosting BCG during the first three days post-immunization, and they can not only host BCG, but they can also do the trafficking of BCG to the draining lymph nodes. And then there are some discrepancies regarding the role, the putative migratory capacity of other cell populations, such as macrophages or monocytes, and especially during the early responses. This is more debate. But even with a major role of dendritic cells, other populations might also be involved and may have different contributions. So there are different ways to improve, regardless which is what are the populations that are involved. There are different ways to improve the capacity of the cells to migrate, and one of them is through fostering their metabolism. And then this is when immune metabolism came into play, and we, our team, have a lot of experience in working in immune metabolism, specifically in TB. And it is well known that metabolic pathways are very linked to the effector functions of immune cells, and also can define their migratory capacity. For instance, it is well known that many glycolytic enzymes are associated and can be preferentially located within the cytoskeleton, or in relationship with the cytoskeleton. And this preferential location not only can modify the enzymatic activity of the glycolytic enzymes, but can also affect the architecture of the cytoskeleton, influencing cell motility. So we have recently evaluated about the impact of human-derived dendritic cells, monocyte-derived dendritic cells, how their metabolism was affected when they were infected with Mycobacterium tuberculosis. And what we found is that either if we infected these cells with Mycobacterium tuberculosis or with irradiated MTV, we observed that there was a clear shift towards glycolysis, that it can be evidenced by the increase in the release of lactate, the increase in the uptake of glucose, the increase in the expression of glycolytic genes, and also the increase in the production of ATP that is associated to glycolysis. And we also observed that there is an increase in the expression of a very important transcription factor, which is HIF-1 alpha, that can also lead to the upregulation of glycolytic genes. And we also demonstrated that if we block the activity of HIF-1 alpha, then we can impair the increase in the glycolysis in these dendritic cells. So we wonder what happened in terms of the functionality of these cells if we blocked HIF-1 alpha in these dendritic cells. So first of all, what we found is that in terms of the ability of these dendritic cells to polarize CD4 T cells in vitro, we didn't observe any impact at all. So dendritic cells in which HIF-1 alpha was inhibited, there was no impact in their capacity to polarize CD4 T cells. But this is what is happening in vitro. So for the dendritic cell to be able to activate CD4 T cells in an in vivo context, it first needs to migrate to lymph nodes. So that is why we wonder, what about the migratory capacity of the cells? And we found that if we blocked HIF-1 alpha in these dendritic cells, or even if we just blocked the conversion of pyruvate to lactate, I mean the production of lactate, we could block the chemotactic ability of these dendritic cells, also their migratory ability in vitro and also in vivo. So we could basically recover less cells from the lymph nodes in vivo also. So that means that HIF-1-alpha-mediated glycolysis is required to trigger the migration of these tuberculosis-stimulated dendritic cells into lymph nodes. So after that, we decided to evaluate if this also applies in the case of BCG-infected dendritic cells. And what we use in this case is bone marrow-derived dendritic cells, and we infect them with BCG, and we observed that in this case, the infection with BCG increased the expression of HIF-1-alpha. These cells now also secrete lactate. They show an increased glycolytic activity. When we blocked HIF-1-alpha, again, the secretion of lactate was reduced. The migratory capacity in vitro was reduced, and also the migratory capacity in vivo was also reduced. But in this case, these are dendritic cells that are infected with BCG. So HIF-1-alpha is necessary also for the efficient migration, in this case of BCG-infected dendritic cells. And also we observed that it has an impact in the activation of the adaptive immune response, because we observed that the animals that receive dendritic cells infected with BCG, but in which HIF-1-alpha was inhibited, in these animals we observed that the generation of interferon-gamma producing CD4 cells was limited. So based on all this, we propose the following hypothesis for our project, which is that by stabilizing HIF-1-alpha in BCG-infected dendritic cells, we might enhance the trafficking of BCG to the lymph nodes, thereby facilitating the early onset of the adaptive immune response and potentially leading to a prolonged protective immunity. So this is the hypothesis behind the proposal. And so we propose to use two drugs to modulate HIF-1-alpha during the immunization, which are these ones, the MOC and Tilorone. So the first one, the MOC, it's a drug that can be used to stabilize HIF-1-alpha levels by inhibiting its degradation. And we have largely used this MOC in our group to stabilize, for instance, to stabilize HIF-1-alpha in macrophages. Also, we have used it to decrease bacillary loads in tuberculosis-infected animals. And we have also used it to recover the chemotactic ability of dendritic cells that comes from monocytes from TB patients. So this drug can be used being administered intraperitoneally and have been used mostly in preclinical models. But to increase the chances of success of our project, we also propose to use a second drug, which is Tilorone, which uses a completely different mechanism of action to increase the activity of HIF-1-alpha. In this case, Tilorone was also shown to induce a metabolic shift in macrophages and also to reduce bacillary loads. It can be used, it can be administered orally, and it is used in clinics in certain countries as an antiviral drug. So the objectives of our project, the aim is to demonstrate that the timing of the onset of the adaptive immune response from BCG vaccination influence the level of protection against MTB, and we propose two objectives. The first one is to define the experimental mirroring model of BCG immunization that optimize HIF-1-alpha modulation to accelerate the onset of the adaptive immune response by measuring both the kinetics of BCG arrival and the presence of polyfunctional CD4 T cells in the draining lymph nodes, and we will also explore the spleens at day 3 and 5 post-BCG. So this is very early stages post-immunization, and we will do it with the contribution of our validate partners from Argentina, from the INTA Institute, Fabiana Vichy and Federico Blanco. And our second objective is to determine the impact of this modulation on HIF-1 alpha during the immunization on the levels of protection. And this will be carried out with the collaboration of Marcelena Otamayo and Tarudut from the Colorado State University in the USA. So for the first objective, we propose two different approaches. In the first one, the drugs will be administered at systemic levels, and with the idea of this way of administration is to potentially enhance the migratory ability of those populations that are involved in the actual transporting of BCG to the draining lymph nodes. And compared to the second one, we expect we expect that the drugs will be able to have a prolonged effect. And in the case of styrorone, the enucleation pathway can have a translational potential. But of course, the downside is that the compounds will also affect other cell types. But to mitigate this downside, we propose the second model, which of course has also its own limitations. But the second model involves the immunization of mice with BCG-infected bone marrow-derived dendritic cells that will be derived from CD45.1 mice that will be pretreated or not with the drugs. So in this model, everything is more under control. We will know which are the cells that will be the ones carrying BCG at least from the beginning, and the cells will be traceable. So this model is useful for mechanistic studies, and of course the downside in this case is that this population bone marrow-derived dendritic cells may not accurately replicate the relevant population that is involved during dermal inoculation. So these are the readouts for the first objective. At day 3 and 5, we will collect cervical lymph nodes and spleens, and we will use also BCG that is recombinant for GSP, and we hope that through FACS and CFU, we will have an idea of whether which are the cells that can host BCG in the lymph nodes, and whether we can recover live BCG or not from them. And then we will do a battery of assays to have an idea of about the quality and magnitude of the adaptive immune response that is induced, such as measuring the level of cytokines and to characterize the polyfunctionality of T cells. And then we will allocate some splenocytes to evaluate the capacity of the cells to inhibit MTB growth. Yeah, I'm about to finish, just 2 slides. The second objective is about the long-term protection. So the idea is to measure after challenge at 2 time points to determine bacillary loads and perform histopathological studies. I'm going to finish because I'm over time. So the expected outcomes from this project will be to gain a better understanding of the initial interactions between the host and the pathogen that shape the antimicrobial tuberculosis immune response, to generate initial data on whether accelerated adaptive immune response can enhance MTB protection, and to contribute to the characterization of lymph node cell population that harbor BCG following immunization. Again, just to thank the collaborators, and especially to really thank Validate Leadership and sponsors for making all these amazing opportunities available and accessible for all of us. Thank you.
Thank you very much, Luciana. Questions, please? Got one here and then one there.
Hi, I really enjoyed that. That's beautiful work. I imagine you're also going to factor chemo attraction by looking at what chemokines particularly could actually be really are triggered in the modulation of, for instance, you mentioned CCL21 earlier on. Do you intend in your plan for experiments to look at chemokines that might be helping migrate mature dendritic cells to the site of our lymph nodes, the site of presentation. And also at the beginning you mentioned that your aim, or the aim, I mean ideally, is to really have a quicker way of presentation of BCG to the lymph nodes. Is there a time factor that you think actually with modulation you can actually factor into an experimentation?
Okay, let me see if I understood. So the first question was about whether HIF-1 alpha could also affect the presentation?
Yeah, so in antigen, in migration of dendritic cells to the site of antigen presentation, which is lymph nodes to T cells, I wanted to check whether you're considering looking at what chemokines are involved in helping chemo attract or movements of disease to the site.
Well, for this first set of data that we will collect, I mean, we will have maybe the samples to then maybe evaluate chemokines, but we don't foresee as the first in first place to measure chemokines, but it could be interesting to do it. Yes, maybe it can worth it, but it's not still planned this way, but it can be worth it, really. Especially because one of the hypotheses is that maybe some of these treatments may also affect the quality of the populations that may be that might travel and might harbor BCG to the draining lymph nodes. So maybe the different signs can also call different populations, and they may also affect the final outcome that we are seeing.
Okay, thank you very much, Luciana.
Yeah.
Well done.
Okay, thank you.